Welcome everyone. Today we will explore in vitro in vivo correlation, or IVIVC. In this presentation, our focus will be on mechanistic PBPK-based IVIVC and how this approach enhances physiological accuracy compared to traditional deconvolution methods. We'll cover the different IVIVC levels A, B, C, multiple levels C and D, with key concepts crucial for understanding its application. There are two approaches to develop an IVIVC: direct one-stage, convolution-based approach and sequential two-stage, deconvolution-based approach. In this presentation we will focus on the two-step, deconvolute and then correlate, workflow. Additionally, we'll explore both internal and external model validation strategies that ensure the robustness of our predictions. Finally, we'll discuss the regulatory applications, particularly how virtual bioequivalence (VBE) trials can predict formulation success, providing a significant advantage in drug development. Let's get started. IVIVC is a predictive mathematical framework that describes the relationship between an in vitro property such as dissolution or release rate of an extended release dosage form and its corresponding in vivo pharmacokinetic response, such as plasma drug concentration or the amount of drug absorbed. Specifically, IVIVC links how a drug dissolves in a laboratory setting to how it behaves in the human body. Now, let's talk about why it actually matters. What's the real purpose behind establishing these correlations? A validated IVIVC reduces regulatory burden. It can serve as a surrogate for in vivo human bioequivalence studies through bio-waiver applications for certain SUPAC scale-up and post-approval manufacturing or formulation changes. It lowers clinical trial cost burden, saves significant time and money by replacing expensive, resource-intensive human bioequivalence trials with predictive laboratory testing. Guides dissolution method development by establishing bio-relevant testing conditions that accurately reflect deconvoluted in vivo drug performance. It establishes clinically meaningful dissolution specifications that allow acceptable formulation or manufacturing variability without compromising overall bioequivalence. It guides formulation selection by identifying formulations whose in vitro dissolution profiles best correlate with deconvoluted in vivo profiles. Let's explore how different levels of IVIVC vary in complexity and application. Level A IVIVC is the gold standard for regulatory submissions and bio-waivers. It is highest level, point-to-point correlation between in vitro dissolution and in vivo dissolution, or percent bioavailability, across entire time course. It could be linear or nonlinear correlation. Level B uses statistical moment analysis. It compares the mean in vitro dissolution time versus mean in vivo dissolution time or mean in vivo residence time, MRT. While useful, it does not uniquely reflect the plasma profile, as different in vivo curves can yield the same MRT value. Level C correlates a single dissolution parameter, such as time to 50 or 90% dissolution, with a single pharmacokinetic parameter such as Cmax or AUC. It is the weakest level of IVIVC and is of limited regulatory utility unless multiple level C correlations, at several dissolution time points, can be established. However, in such cases, level A may also be feasible. Level D correlation is a qualitative and rank order, non-mathematical, correlation that compares the in vitro release data with in vivo absorption profiles. It ranks formulations by availability of drug or release rate, that is, fast, medium, slow release, to match general absorption trends seen in subjects. Level D IVIVC is not considered a formal correlation and lacks official standing in regulatory guidelines. To develop a robust level A IVIVC, we need specific inputs. First, we require in vitro dissolution profiles from 3 formulation batches exhibiting slow, medium, and fast release data using compendile methods. Then, we need in vivo plasma concentration time profiles from same above batches. We also need reference in vivo plasma concentration time data, obtained following an API solution, immediate release formulation, or intravenous administration. Why is this so important? It serves as our biological baseline to fix drugs' disposition parameters such as clearance, volume of distribution and effective permeability. Fixing these disposition parameters ensure the IVIVC reflects formulation release behavior rather than underlying physiological variability. There are two approaches, one-stage versus two-stage IVIVC. One-stage method is a convolution-based approach. It directly links the in vitro dissolution and plasma concentration data using the convolution integral. This approach avoids any intermediate step errors or the need for an immediate release or intravenous reference dose. In contrast, sequential two-stage method is the deconvolution-based approach, which involves two steps. First is the deconvolution of plasma profile to obtain in vivo dissolution or absorption profile, and the second step is establishing the correlation between in vivo and in vitro dissolution data. Key difference between these two approaches is the trade-off between power versus simplicity. One stage offers superior statistical power and parameter accuracy but requires complex nonlinear modeling, making interpretation, troubleshooting, validation, and regulatory communication more challenging. Errors may propagate between stages, but the two-stage approach is simpler, easier to model and validate, and thus regulatory friendly. In this presentation we will focus on the two-stage IVIVC. To establish a two-stage IVIVC, the first question is how can we derive the in vivo input rate from plasma concentration time data? This starts with deconvolution. Deconvolution is the mathematical process of deriving an in vivo dissolution or absorption profile from the observed plasma concentration time data. To do so, we have traditional and mechanistic deconvolution methods that differ in how they separate drug input from body disposition effects. Traditional IVIVC uses mathematical models such as Wagner-Nelson, Liu-Riegelmann, or numerical deconvolution to estimate overall systemic bioavailability, not the true in vivo dissolution. While mechanistic deconvolution applies PBPK-ACAT frameworks to separate true in vivo dissolution from absorption barriers and hepatic, intestinal first-pass metabolism. It addresses complex ADME behaviors beyond capabilities of traditional deconvolution. Let's explore the traditional methods and their limitations first. Traditional deconvolution approaches fall into two categories: model-dependent and model-independent methods. Model-dependent methods like Wagner-Nelson and Liu-Riegelmann use mass balance equations across pharmacokinetic compartments to estimate the fraction absorbed over time. Model-independent methods use numerical deconvolution. Numerical deconvolution works backward. It subtracts the body's disposition, or unit impulse response UIR, from the observed plasma concentration time profile to calculate the unknown in vivo drug dissolution or absorption rate. However, these traditional approaches have significant limitations. They estimate systemic bioavailability rather than true in vivo dissolution. They do not tell whether reduced bioavailability is due to incomplete absorption or significant first-pass metabolism in the gut or liver. They assume simple compartment model with first-order absorption and ignore nonlinear absorption or clearance processes due to saturable transporters or enzymes. They assume the terminal phase of the plasma profile is independent of ongoing absorption, which may not hold true if drug is still being absorbed in the colon. Those are some pretty significant limitations. Mechanistic deconvolution addresses these limitations. Traditional IVIVC estimates cumulative bioavailability, not the true in vivo dissolution time profile. It combines dissolution, gastrointestinal absorption, enterocyte binding, intestinal and hepatic first-pass metabolism, and transporter effects into a single bioavailability parameter. Whereas mechanistic deconvolution gives us the granularity to understand each step independently. It separates all these individual processes involved in drug absorption. GastroPlus uses a CAT or Advanced Compartmental Absorption and Transit model to provide several distinct outputs like total drug dissolved representing true in vivo dissolution, total drug absorbed representing segment by segment gastrointestinal absorption, amount reaching the portal vein before hepatic first pass, and the final systemic exposure or percent bioavailability. Now, let us compare the input requirements for traditional versus mechanistic deconvolution side by side. Traditional deconvolution requires only the plasma concentration time profile and dose. It is empirical method and does not account for the drug's physiochemical, biopharmaceutical properties or body's physiological details. For PK disposition, traditional deconvolution assume a simple linear 1, 2 or 3 compartment open model with first-order absorption. The key limitation of traditional methods is they cannot handle saturable or nonlinear disposition. It is a real concern when the drug is a substrate for metabolic enzymes or efflux transporters. Mechanistic deconvolution using PBPK-ACAT modeling requires significantly more information but yields far richer insights. It requires drugs' intrinsic physiochemical properties, such as molecular weight, log P and pKa, biopharmaceutical properties such as pH solubility, effective permeability, pH, and particle size distribution. The ACAT model incorporates detailed human GI tract physiology, including regional pH values, segment dimensions, transit times, and bile salt concentrations. Mechanistic deconvolution integrates PBPK, ACAT models to incorporate nonlinear Michaelis-Menten kinetics, complex ADME processes, including transporter dynamics across the gut wall and liver, first-pass extraction, FPE, plasma protein binding and blood-to-plasma ratio to allow accurate deconvolution under saturable clearance and nonlinear disposition. Now let us look at the output differences. Traditional deconvolution produces a plot of fraction absorbed or systemic bioavailability over time, essentially the amount reaching the central compartment. It provides no regional absorption insights. It cannot distinguish whether low bioavailability is due to incomplete absorption or due to first-pass losses. Thereby, traditional deconvolution functions as a black box, with no mechanistic understanding of the underlying ADME processes. Mechanistic deconvolution, on the other hand, produces a true in vivo dissolution versus time profile along the gut. It describes site-specific absorption across different GI tract segments, separately quantifies gut and liver contributions to first-pass extraction. Thus, mechanistic deconvolution fully separates all mechanistic processes, giving us distinct outputs, like total drug dissolved representing true in vivo dissolution, total drug absorbed representing segment-by-segment absorption in GIT, amount reaching the portal vein before hepatic first pass, and the final systemic exposure or percent bioavailability. This level of detail is what makes mechanistic IVIVC so powerful for formulation development and regulatory strategy. With that comparison fresh in mind, let's put it all together and walk through the development of a two-stage mechanistic IVIVC. The development of IVIVC follows a systematic two-step process. In Step 1, deconvolution, we derive the in vivo input rate from plasma concentration time data. For traditional IVIVC, it gives us the fraction absorbed in systemic circulation or F%. For mechanistic IVIVC, this step gives the true in vivo dissolution time profiles. In Step 2, correlation, we establish a mathematical relation or an IVIVC correlation function between in vitro dissolution data and in vivo input rate derived in Step 1. This mathematical relationship may be either linear or nonlinear, and a single IVIVC correlation function should remain valid across multiple formulations with varying release rates, such as low, medium and high release rates. Now, the IVIVC correlation function established in Step 2 can be applied to predict plasma concentration time profiles of drug formulations using their dissolution profiles. Let us now walk through each step in detail, starting with Step 1, the deconvolution process. Step 1 is deconvolution, in which plasma concentration time data are converted into the in vivo dissolution in the mechanistic IVIVC, or the fraction absorbed in the traditional IVIVC. Traditional IVIVC uses Wagner-Nelson, Liu-Riegelmann, or numerical methods, while mechanistic IVIVC applies PBPK, ACAT models. The dissolution profiles are optimized by converting the discrete dissolution data points into a smooth and continuous release curve using single, double, or triple Weibull functions to accommodate a wide variety of release profile shapes, from simple monotonic release to complex biphasic or triphasic patterns. When traditional IVIVC fails, mechanistic IVIVC using Weibull functions may succeed by capturing true in vivo dissolution rather than lumped absorption. Now, we will move to Step 2. To find one single correlation function between in vitro and in vivo dissolution. Step 2 is establishing the correlation between in vitro dissolution and the deconvoluted in vivo dissolution profiles. The goal is to find a single, universal correlation function that works across all slow, medium, and fast release formulations used in the IVIVC development. This requirement gives the IVIVC its predictive power for new formulations. Several correlation functions are available, including linear, power function, quadratic polynomial, cubic polynomial, and time shift or timescale transformations. The choice of function is guided by selection criteria such as the Akaika Information Criterion, or AIC, which balances goodness of fit with model parsimony. With our correlation function established, we're ready for the payoff. In the convolution step, where we will use this universal correlation function to predict plasma profiles from dissolution data alone. Convolution derives plasma concentration time profiles from in vivo dissolution using either linear compartmental PK models or mechanistic PBPK-based models. Linear convolution uses mathematical superposition and a compartmental PK-derived unit impulse response, UIR, to predict plasma concentration time profiles. In mechanistic convolution, such as within GastroPlus or PKSim, a separate UIR is not required. The in vivo dissolution profile is directly integrated into PBPK-based physiological differential equations, incorporating GI transit, regional effective permeability, PEF, tissue volumes, and clearance. The plasma concentration profile is then generated by solving these differential equations iteratively over time. Convolution represents the core strength of IVIVC. It predicts plasma concentration time profiles for new formulations and evaluates scale-up and post-approval SU-PAC changes without new clinical trials to enable regulatory bio-waivers and faster formulation development. A model's strength relies entirely on its validation. Let's now see how we confirm the predictive performance of our IVIVC model. An IVIVC model requires validation before use for regulatory purposes. The validation workflow follows 3 steps: correlation, convolution, and comparison. In correlation, the developed IVIVC equation is first applied to in vitro dissolution data to calculate the corresponding in vivo dissolution profiles. It is important to note that these calculated in vivo dissolution profiles, generated from in vitro dissolution using the IVIVC correlation equation, are not the same as the fitted or deconvoluted in vivo dissolution profiles obtained from plasma concentration time data. Next, in convolution step, we input the calculated in vivo dissolution profile into the PBPK simulation to generate predicted plasma concentration time profiles. Now, we compare the predicted profiles against the observed clinical data using the % prediction error metric. This comparison tells us how accurately the IVIVC can predict real-world pharmacokinetic behavior, the ultimate test of the model's utility. There are actually 2 flavors of validation, internal and external, and they serve very different purposes. Validation can be internal or external, and both serve different purposes. Internal validation uses the same formulations that were used to develop the IVIVC. It checks model consistency and robustness, meaning, it checks if the developed IVIVC model can reproduce the data it was built from. The acceptance criteria for internal validation are strict. The mean % prediction error must be 10% or less, and individual formulation prediction errors must be 15% or less. External validation is a stronger test. It uses formulations that were not part of the IVIVC development. It tests the model's predictive capability using truly independent data. The acceptance criterion is a mean % prediction error of 10% or less. A good internal validation confirms the model is reliable. A good external validation confirms it is genuinely predictive. Once we have a validated IVIVC, one of the most exciting applications is running virtual bioequivalence trials. Let's see how that works. Virtual bioequivalence (VBE) trials are a powerful application of validated IVIVC and PBPK modeling. VBE uses computer simulations to demonstrate bioequivalence, that is, they assess whether a test formulation matches the rate and extent of absorption of a reference formulation, much before conducting actual human studies. In bioequivalence study, we must demonstrate that 90% confidence interval for geometric mean ratios between a test, or generic, and reference, or brand name, product falls within 80 to 125% for key pharmacokinetic parameters like Cmax and AUC, under both fasted and fed conditions. VBE trials reduce costs, shorten timelines, minimize clinical testing, and support regulatory approval of generic drugs. Now, let's see the steps in VBE in silico approach. First, we define population parameters such as clearance and volume of distribution to capture real-world physiological variability. Next, we select an optimal number of virtual subjects to balance statistical power, time, and cost. If there are too few subjects, it may fail B trial even when the product is truly bioequivalent. Too many subjects can result in wasted time and resources. Multiple virtual crossover trials are then performed to compare test and reference formulations under identical crossover conditions. followed by analysis of geometric mean ratios of Cmax and AUC at 90% confidence intervals to assess bioequivalence. While not a replacement for actual clinical trials, VBE can predict whether a formulation is likely to pass or fail. It provides an essential decision-making tool to de-risk formulation development and optimize clinical study design before committing to expensive human trials. And that brings us to the end of this presentation. Here are a few key references supporting these IVIVC concepts discussed in this presentation. I hope this presentation has provided a better understanding of Mechanistic PBPK-based IVIVC, detailing the two-step workflow for two-stage IVIVC development, model validation techniques, and the use of virtual bioequivalence trials to predict regulatory success in drug development. I would be glad to address any further questions you may have. Thank you for your attention.